The enzyme that I studied this semester is called gamma-secretase. So what exactly is gamma-secretase? Gamma-secretase can be thought of as molecular scissors. It's an intramembrane protein involved in the proteolytic cleavage of proteins within the cell membrane. And what this does is the cleavage often activates some type of molecular mechanism. So here is the actual structure of gamma-secretase seen on the left. It's an intramembrane hydrolase. It's overall hydrophobic, and it has 4 different components. But the most important one that I'll be talking about today is the presenilin component, as this is the catalytic component that holds 2 aspartyl residues that are activated by water. And that's seen here in this very simple illustration of the activation. So gamma-secretase is involved in something called Notch signaling, so a signaling pathway. And here you can see these scissors are representing gamma-secretase. And essentially it cleaves this protein, and this intracellular domain affects gene expression overall. And this is really important because Notch signaling is very important for cell-fate decisions during development, as well as stem cell upkeep in adults. And altered levels have been associated with many different cancers. Gamma-secretase is also involved in Alzheimer's disease, specifically in the cleavage of the APP protein into the amyloid beta protein. And these amyloid beta proteins stick together and make these neurogenic plaques that are characteristic of Alzheimer's disease and essentially cause the brain tissue to die, as you can see in the picture below. So for this reason, gamma-secretase is a pharmaceutical target. There have been gamma-secretase inhibitors used. However, they can disturb other pathways such as Notch signaling, and this was actually seen in a clinical trial. There's also gamma-secretase modulators, which are often a little bit more specific in their target and help to minimize disturbing other molecular pathways. So who studies gamma-secretase? Our first person is Michael S. Wolf, a professor of medicinal chemistry at the University of Kansas. Some of his contributions that have been important are some contributions to the structure, specifically the fact that it involves 4 different parts, and that the protein has 4 different parts, and that the presenilin component is the part that's actually catalytic. He's also studied familial Alzheimer's disease, and he's studied how these mutations that cause hereditary Alzheimer's disease affect the processing of the APP protein. And the future goals of his lab is to develop probes for the future therapies and to further understand the substrate recognition of gamma-secretase, and then to also keep on understanding how different mutations cause differences in gamma-secretase function. The next person is Bart de Strooper, who is an Alzheimer's researcher on the molecular level of things, and he's a director at the UK Dementia Research Institute. So something that he's currently looking at is the involvement of microglial cells in Alzheimer's disease, as these are linked to neuroinflammation and the clearance of those neurogenic plaques. But an issue is that in the mouse models, microglial cells are very different than what we see in humans, so which makes it much harder to study. But Strooper's team has actually developed a chimeric model of the human microglial stem cells, injected them into a brain of mice, and was able to see very similar cells from the human to the mouse. And these cells also retained their human transcriptome, which will be very useful for future microglial study cells of Alzheimer's disease. So I looked at a study that really tried to understand the structure of gamma-secretase. And just as a reminder, gamma-secretase is cleaving its substrate in this hydrophobic environment of the cell membrane, but yet it's activated by water. So this study kind of sought to understand biochemically how this was happening. And what they suspected was that there's this hydrophilic water pocket that's present within the membrane. So to better understand the structure of the protein, they use cysteine scanning mutagenesis. And essentially, this allows them to use EasyLink biotin HDPD protein. And what this does is it binds to the sulfhydryl groups of those cysteines by making a disulfide link. And if they are labeled, then we know that those sulfhydryl groups are free and therefore can act with water, and they're not interacting with a therefore different part of the protein or something like that. So they essentially chose some residues that they thought would possibly be involved in this pocket within the protein that can hold water. So on the left side, you can see there was a Western blot to see what was actually labeled. You have the control group and then several residues that were mutated to cysteine. And then the last lane is just wild type. And then on your vertical axis, you have all the antibodies. Nicastrin was the negative control. And this is just a different part of the actual enzyme. Here are the rest of the labels, the full length, the N term, and the C term of the protein, and the positive control, which is the PEN2. And the results was that they only found one residue that they suspected to have this free sulfhydryl group within the N terminus. So even though these results were not as anticipated, they thought that there was possibly some type of steric hindrance for the biotin to mark the free sulfhydryl groups, because if there is this pocket holding water, it's going to be in the interior of the protein. Although these results were not as anticipated, the data did eventually lead the researchers to another experiment that biochemically proved the catalytic aspartate was present in a water cavity. And this was the first biochemical piece of data outside modeling and speculation that there was a water cavity present in gamma-secretase as gamma-secretase. So for future directions, I think it's important to really get a good hold of the structure and function of gamma-secretase so that different gamma-secretase inhibitors or modulators can continue to be examined to understand how they can make treatments. And one way of finding treatment is by looking at the presenilin extracellular domain. So previous research has shown that this might be the extracellular domain of the catalytic component might be involved in activating gamma-secretase. So antibodies can be made for this region to help lower its activity, gamma-secretase. So one way to do this is with site-directed mutagenesis by directing them to an alanine substitution of the extracellular domain, incubating them with a cell line, lysing them, and measuring the Notch intracellular domain to measure the gamma-secretase activity. And the reason I say Notch intracellular domain is because it's hard to have an in vitro study of Alzheimer's cells. And then mass spectrometry can be used to measure how much of the NICD species is present in the cell lysate, and then we can compare this to the control, which would be a wild type, the positive, and then the negative control can be gamma-secretase with a known inhibitor. And this would just give us more insight if maybe the extracellular domain is an important part for possible treatments. Thanks for listening.